A very warm welcome to the participants of the Proteomics Advanced Winter School 2021. Presented by Proteomics Lab, Indian Institute of Technology, Bombay. Day 5. Quantitative Proteomics, Label-Based. Lecture 4. Human Proteome Atlas.
Good morning, ladies and gentlemen. We are extremely delighted to welcome Professor Matthias Ullin. Professor Ullin received his PhD at Royal Institute of Technology, Stockholm, Sweden in 1984. After a postdoc period at EMBL Heidelberg, Germany, he became professor in microbiology at KTH in 1988. His research is focused on protein science, antibody engineering, precision medicine, and ranges from basic research in human and microbiology to more applied research including clinical applications in cancer, infectious diseases, cardiovascular diseases, autoimmune diseases, neurobiology. His research has resulted in more than 650 publications. His group was first to describe a number of innovations in science including engineered protein A and protein G for purification of antibodies, affinity tag for purification of recombinant fusion proteins, solid phase method based on magnetic beads for DNA handling using the biotin stuffed evidence system, pyrosequencing leading to first next generation DNA sequencing instrument 454 DROSH, Affibodies, protein binders aimed for therapeutic application. Since 2003, he has led an international effort to systematically map the human proteome and transcriptome to create a human protein atlas using antibodies and various omics technologies. This effort has so far resulted in 5 major launches, all published in the journal Science. The Tissue Atlas showing the distribution of proteins across human tissues and organs, the Cell Atlas showing the subcellular location of human proteins in single cells, the Pathology Atlas showing how cancer patients' survival is tied to RNA and protein level, the Blood Atlas showing the expression of proteins in human immune cells and also mapping the proteins in human blood, the Brain Atlas showing the proteins expressed in different parts of human, pig and mouse breeds. Join me in welcoming our plenary speaker for the day, Professor Matthias Ullin.
We can hear him. Yeah, we can hear you. All of us can hear you. I have shared my screen on Teams. Yeah, you're good to go.
So I started by just showing that 20 years ago, the announcement of the human genome gave us a blueprint for human biology and diseases. But obviously, in order to study the future of biology, we have to study the proteins, and that's why you are on this workshop. So what we asked us 20 years ago, what are the building blocks of cells, tissues, and organs? And really, what we want to do in the end is, can we model human biology from its protein building blocks? And this is what we're trying to do. So I am the founding director of an institute in Stockholm called Science for Life Laboratory, which is a national infrastructure for life science. And this was started in 2010 to actually do big data analysis and technology-based infrastructure. We are now about 1,200 researchers, and last year we ran about 2,000 projects. Before I continue, I just want to say that I'm co-founder of several startups, and I will mention some of them in this talk, especially Antibody-pedia, but I'm also doing collaborations with AstraZeneca. So what I thought I will do is to give you a little bit about the 20 years with the Protein Atlas, the new version 20, talk about precision medicine, and then talk about the movies that we made, which are journeys into the human body. So the Human Protein Atlas, the vision is to map all the human proteins using technologies to create an open access atlas of the human proteome for researchers in academia and industry. This is very much a European effort from Sweden, but we have very important collaborations with people in South Korea, BGI, but also the most important collaborator has been in India, in Mumbai, where and I will come back to this. So in Science magazine, some months ago, we published a digital booklet that takes you through the 20 years of HPA and the milestones. This is a 140-page document, so I'm not going to go into it in detail, just show you some of the highlights in this. One of them, it started back in 2000 with a chromosome 21 pilot. We decided to make antibodies to all proteins encoded from chromosome 21, and then to analyze protein profiles in tissues. 5 years later, we launched the first protein atlas with 7,000 antibodies, and we were very proud to have 400,000 immunohistochemistry images in that launch. But then we started a collaboration with Sanjay Navani and colleagues in Mumbai, and we set up then a site in Mumbai, in central Mumbai, for high quality pathology annotations. And this has been a fantastic collaborations between the HPA and the Indian site. And the site in India has annotated more than 10 million pathology images. And I think that's the largest number of annotations in the world. In 5 years later, 2015, we published the first major paper in Science, the Tissue Atlas, and it is actually one of the most cited research publications from Europe in the last 6 years now. So we're very proud of that. But since then, we have published quite a few additions to the Protein Atlas, a subcellular atlas in 2017, a cancer atlas in 2017, a blood atlas about 2 years ago, well, actually 1 year ago, and then a brain atlas in April last year, and a metabolic atlas. And in total, more than 550 publications from our group. So what have we achieved then? We have generated ourselves about 55,000 antibodies. 21,000 of those has passed the stringent requirements, and we have created more than 10 million immunohistochemistry. And we've done a lot of validation of antibodies, more than 70,000 experiments. So we're very proud to have generated more than 10 million images using these antibodies. And it makes us, I think, the largest effort in the world to generate tissue images. And each one of those images has been annotated by a pathologist in India. So we're very grateful to that. So the data we have in the Protein Atlas now is open access. We have more than 300,000 visits every month. And it is one of the 3 largest biological databases now in terms of visitors in the world, actually. And we're very, very happy for that. So what have we learned? Well, we have learned that about 10% of the proteins are what we call housekeeping, and thus needed in all dividing cells. About 15% of the proteins are specific, and present in only one or very few tissues. And about 13% of the proteins are secreted. We call it the human secretome. And these are, of course, very interesting for all of us that are looking at blood proteins. We also made a lot of movies, and I thought I would show you some of these, and I hope that the sound actually comes through. And the first one I want to show you is a movie. And we have made this using a new technology called iDisco, using antibodies. So these are real images inside the human body. And we're going to start with a beautiful, stunning movie of the inner ear of humans.
This is the inner ear. It is one of the smallest, yet one of the most important organs in our bodies. The functions of the inner ear are multiple. It consists of the cochlea, responsible for our hearing, and the vestibular system, responsible for our balance. Damage to the inner ear can not only lead to a loss of hearing, but also imbalance, tinnitus, and vision impairment. With these 3D images, it's possible to really appreciate the beauty and complexity of the inner ear.
So I just wondered, could you hear the sound of the video?
Yes.
Okay.
Yes.
Well,
Yes.
yeah, very good. Thank you very much. So a few months ago, in November 19, we released the version 20 of the video. We had two major new parts of it: a single cell type atlas and a prefrontal cortex atlas. And I want to quickly go through that. So we have used then the RNA analysis and the new single cell analysis from human tissues to try to integrate that with antibody-based profiling. And we have been looking at 192 single cell types and actually analyzed what proteins and RNA are in these cell types. And just to show you one example, this is the PSA. You can then get these kind of clusters. We call it, this is the prostate, and every point here is a single cell, and then it says how much in these cells is the PSA expressed, and you can see there are two clusters and they happen to be glandular cells. And then you can actually go in and look at this with the antibody in the protein atlas. And all of this data is available in the Human Protein Atlas. You can also go in and look for the classifications. So here, for example, we are showing all the tissue specific or cell type specific proteins. And we can, for example, see that there are 231 proteins which are specific for early spermatids in the germ cells. And there are 800 proteins which are shared between late and early spermatids. And then you can click on this bubble and then you can see what proteins it is. And I hope that you all can go and do this kind of analysis. It's also interesting to look at proteins which share expression. And for example, we now have the proteins which are shared between ciliated cells, lung, and testis. And this obviously, when you look then in the protein atlas, you can see that this is ciliated cells in these two tissues. So we're trying then to integrate the single cell analysis with antibody-based profiling, and we also do quite a lot of proteomics, mass spectrometry, but I will not talk about that today since the whole workshop is on that. We have then moved on and are now doing a lot of multiplex tissue profiling. So we're combining single cell data with the multiplex in immunohistochemistry. And just to show you the beauty of this, I show you a kidney stained with 6 antibodies. And you can clearly see then the glomeruli here in sort of red. You can also see here both the proximal tubuli, the distal tubuli, and the collecting ducts in green, and you can see very, very nicely where your protein is localized. We also are working a lot now with detail of the human brain. And what we've started with is the prefrontal cortex, which is a very important part of the human brain. And I'm not going to go into the details here. This is people at the Karolinska Institute. But we are now creating a very detailed molecular map of all the different regions of the prefrontal cortex, but I don't have time to go into that. But what we're doing is do microdissection of brain regions using a punch needle approach. What we're also doing now is to look and do precision medicine. And the idea here obviously is to get the right treatment to the right patients, to do more targeted treatments and better diagnostic methods. The main way of doing that is we're using targeted proteomics, we're using mass spectrometry, but we're also doing it using antibodies, and we are using now a very new technology called Olink Explorer that allows us with very low sample volumes, 5 microliters of blood, to have a look at about 1,500 targets with very high sensitivity. We can go down to cytokine levels, and this we can do in one multiplex assay. So this is quite a fantastic technology, and we hope to have a paper coming out very soon in Nature Communications. So with that technology, then we have then looked at different patient cohorts, and this just shows you the Swedish Scapis Scilab Lab Wellness Profiling Program, where 100 individuals has been followed for 2 years. We're taking samples every 3 months, and then we analyze them with whole genome sequencing, Olink Explorer, targeted proteomics using mass spectrometry, immune cell profiling, metabolomics, again using mass spectrometry, gut microbiota, and so on. So these 100 individuals, I think, is the best studied individuals in the world, actually. So what you can then do is that you can look at the protein levels. In the middle here, you see the famous leptin gene or protein in the human plasma. And here you can see in gray, you can see the individual, how they are, the concentration in the blood of individuals. The average for females is here in blue, and the male here. And you can see very clearly then that females have higher levels, but also very interesting is that the individual levels vary quite a lot, both in the female and male, but they are stable during these 2 years that we have sort of studied it. So there is large individual variations, but the actual individual variations are stable during 2 years. And this is, of course, very, very interesting for all of us. You can also do this kind of dynamic UMA plots. Each dot here is an individual, female in red and male in green. And this is then how the whole profile of the proteome is changing during 2 years. And you can see for most of the individuals, they have an individual profile, as I said, but it's stable. They are sort of stable during 2 years. But then there are actually some individuals that are changing a little bit during these 2 years. You can also see on this other omics that they are much more variable. So the proteome is quite stable. We've also been doing whole genome sequencing of this, so we can then compare the genetic variants of these of these individuals with the plasma profiling levels. And it is then interesting to see that some proteins are completely determined by genetics. So you have, for example, this famous receptor PDGF. You can see then there is a variant in the gene where you have the CC variants have lower levels than the TT variants, and the heterozygote has about the same. And you can also see that very clearly here. So there are proteins then that you're born with a genetic variant, and that determines your adult levels of that protein in the blood, which is of course very interesting. So we found a lot of these now, we call them PQTLs, where the protein is determined by the genetic variant of the individual. So we can study then this in different ways, and here is an individual that lost 17 kilos in 3 months. And then we looked at all the different proteins that are changing when that happens. And you can see then that a lot of things are happening. Some proteins goes up and some proteins goes down, which is of course very interesting. Here is a protein, here's an individual that got a serious knee infection. And you can see then that the CRP then is coupled to a lot of different proteins. And in blue then you can see this cascade of proteins which are changing when you're getting a knee infection. We've also been studying preterm children, what happens after you're born very early on, and looked at all the proteins in this. And what you can see here is that they are changing very much early on, and this has been published then. We've also been looking what happens to people that are treated, and these is people with type 2 diabetes, and they are treated with metformin, and we are then following what happens when they are treated with metformin, but also which patients are responding to the treatment and not. So the take-home message from this, and I'm sorry that I'm so brief, is that protein profiling, I think, is a very, very important tool for precision medicine. And that's why I think your workshop is very, very important. We have new tools to complement the mass spectrometry and gel-based methods for comprehensive and quantitative protein profiling. We are particularly fond of the Olink technology that comes from Sweden. And then what is interesting then, that each one of you that are listening to this has your own unique protein profile, which is different from all the others. And this is very stable, at least during 2 years. But when you change lifestyle, or when you have are treated with drugs, there are dramatic changes. So this then sort of shows that precision medicine and protein profiling is very, very important. And this is just to show you the people that has been involved in this work. So in closing then, I just wanted to show you some more movies that we have been working on using this single cell 3D imaging of different, with using the iDisco technology. We have these summarized in a microsite, which is on the Protein Atlas. So if you want to, you can then go and look. Here is
The liver
movie from the
produces
liver.
biochemicals necessary for digestion and growth. Here we explore the liver using 3D light sheet microscopy. We are inside one of the portal veins that run in parallel with the hepatic artery and the bile duct. Here the blood flows from the intestine to the liver.
So in conclusion then, what is human life? Well, it is very clear from us and others that human life involves very few protein building blocks. We think about 20,000 protein-coded genes. There are, of course, isoforms and so on, but it is very few. We believe that there is around 10% which are necessary for cell survival, which is means that about 2,000 proteins are necessary. We have about 15%, about 3,000 protein-coded genes which are unique for one cell type or tissues. And many proteins then consequently is found in several cell types and tissues, and it's interesting to understand if they have different function. And what we're trying to do now is to integrate this quantitative omics data to allow modeling of human molecular life. And the question they are asking now, is it actually possible in the future to create a theoretical model for all the components of human cells, tissue, and organs? I think so very much, and we are moving in that direction. So we are not the only ones that are mapping human building blocks. The Allen Brain Atlas in the United States with Microsoft money, the Human Cell Atlas with Facebook money, the Project Baseline funded by Google, the Watson Health by IBM, and so on are actually doing this. And what is interesting is that all these initiatives are sort of funded from the IT sector, which of course is a consequence that a lot of this is data-driven, technology-driven research. So I'm very grateful to the Wallenberg Foundation that has sponsored this project. We have had more than 600 researchers, and quite a few of those comes from India. So I am very grateful, especially to Sanjay Navani, who was leading the Indian effort. And with that, I just want to show you a picture of where we are in Stockholm at the SciLife Lab, which is to the left on this image. And with that, I want to finish by showing a movie from the brain using our antibodies and the iDisco technology. Thank you very much. Well, first, I just want to say that if you want to see these movies, the full-length ones are actually on the Protein Atlas site. You just click Learn and Videos, and there are now actually 13 movies that you can explore yourself.
The brain controls virtually everything required to be human. The primary signaling unit is the neuron, which sends messages via neurotransmitters and receptors. In this video showing the brain in 3D, an example of an uncharacterized receptor, known as an orphan receptor, here shown in red, has been detected exclusively in the thalamus of the brain. Apart from neurons, the brain also contains glial cells, such as astrocytes shown here in green, which support the work of the neuronal cells as well as protecting the brain at the blood-brain barrier. The blood vessels, shown in red, are also crucial in the blood-brain barrier that protects the brain from unwanted chemicals and infections. However, this blood-brain barrier also hinders drugs from entering the brain, making drug development for diseases inside the brain especially difficult.
So with that, I want to thank you for listening. I'm sorry about the technical difficulties. And maybe should I now move back to the Teams if you want to ask some questions, or should I stay on the Zoom? Maybe that's...
I can hear you very well. If you can hear us, can you hear us?
I think.
So then I stay on the, and I stop sharing then. So thank you very much for listening.
So then, can you hear me?
Yes, I can hear you fine.
All right, great. All right, so wonderful. Thank you so much for the wonderful lecture. And I think movies are really mind-blowing. I'm sure all the participants appreciate that, how much effort goes into doing this kind of research. And with antibodies, you are really able to map, the entire detail. And all these are very high quality publications and high quality images generated from it. We're also very happy that Sanjay Navani, who is one of the colleagues locally here in Mumbai, he is a part of one of these initiatives from HPA team. And wish you good luck for competing with so many other big giants. And I'm sure biology team will win the race of mapping all the secrets of life. So with that, let me open up the forum for the audience here. If participants have any questions or comments to ask for Professor Ullen here. Forum is open now. All right, please.
While we're waiting for questions. Yeah, okay.
Hello, sir.
Yeah, Professor Popia, you want to go?
Yeah.
All right, go ahead.
Sir. Okay. Good evening, Professor Mattis. It was wonderful hearing you. And this was, we got to know about this human protein atlas for the first time for me as well. Also, I had one just small query. Is when we're doing the protein profiling for that 100 people that you have actually taken into consideration, so won't the variation in population due to the genetic variation, the ethnicity, won't that be a challenge?
Absolutely. So that's a very interesting question. And obviously, What I think what we are saying in the publications that are coming out right now in Nature Communications, that since every person has its own unique protein profile in the blood, when we are comparing, when we are diagnosing a patient, actually we should not compare the patient with the population, but with itself. So one should have then sort of a fingerprint of the blood that, and then look at what is the differences longitudinally. But of course it is very important to actually then determine now which are the proteins which are determined by genetics and what are the proteins which are determined by environment. And this is why we have spent so much time now to do whole genome sequencing, get all the genetic variants, and then compare the blood proteins with the genetic variants. But I think it's, but it also shows the beauty of protein profiling for precision medicine, I think. Thank you for the question.
Thank you so much, sir, for clearing the point. Thank you.
Dr. Sudarshan, you want to ask a question?
Yes, thank you for giving me opportunity. But it was a very excellent feeling to hear you and your excellent presentation. And particularly knowing about antibodies itself, which was very, very interesting. Because for us in the lab, for one protein, if we are interested in making antibodies against one epitope or two epitope, we struggle a lot. So that's why I was also wondering, how do you tackle this challenge that there are many proteins which are isoform in nature, and against them, how do you go for making antibodies?
Yeah, that's, I actually get that question every time I talk to the Protein Atlas. So the antibodies that we have actually been designed to actually capture as many as possible of the isoforms from a certain gene. So we have not looked at the isoforms at all. We are very aware that a lot of human biology is because of protein isoforms, but the antibodies are not very useful for that. So I think what we have done has to be complemented by proteomics methods that can actually look at isoforms in a better way than we can do. And we are working, we have some collaborations with Matthias Mann, for example, in Germany, where we've been using the antibodies to capture proteins, and then we use mass spectrometry proteomics to look at the isoforms. But in the Protein Atlas, we are sort of looking at the representative proteins from a certain gene and not the isoforms. In summary then, isoforms are of course very, very important, but we have not yet started working. Well, we are not exploring that. That has to be done by sort of more in-depth and more specialized research projects. So thank you for the question.
Good, thank you very much.
Great. One of the participants, Dr. Hemant Gaura, he has posted a question: is the protein mapping being done for all type of cells in body?
So we have in the tissue atlas that was published in Science in 2015, where we are looking at 42 different tissues and organs. In the single cell type atlas that we have released in November, and it's not published yet, we have looked at 192 different cell types, but we have not looked at the cell types in the brain because it's just technically very hard to do analysis. So we are now working very hard to complement the single cell type mapping also for the brain cells. But it's just isolating single cells from the brain is very, very challenging, of course, because of the axons and the dendrites and how in the brain. So this is something that we're working very hard on. So 192 cell types has been mapped, but we expect them to have maybe twice that in a few years. So then we will have a tissue atlas on one hand, and then a cell type atlas on the other hand, that should then be a good knowledge base for everybody working with human biology. Thank you for the question.
That's wonderful. Yeah, great. Argyo, you want to ask a question?
So, sir, basically, like if we are seeing into the domain of biology now, many of the peoples are either moving into biosimilars or something like that. Like if you want to design something unique, such as the antibodies, what you are using in case of Atlas antibodies, like there are series of layers of data analysis and the quality check, like after passing this on criteria only you can release these antibodies for the full-fledged. Like for the researchers like us, can you suggest like what layers of data analysis that one should do before before claiming that antibodies are perfect for that particular tissue or that particular domain?
Yeah, that's a very important question. Thank you. So obviously, since we've been working so hard with antibodies, we also know the difficulties with antibodies. And a very important issue here is what kind of sample you're analyzing. So if you have samples, for example, in therapeutics, you want the antibody to bind to the native protein, obviously, in the human body. And that actually is quite simple. It is actually simpler than doing what we are doing, where we are actually analyzing proteins which have been treated in different ways. So immunohistochemistry, which is the sort of pathology images, you actually treat the sample with usually 120 degrees. So you basically boil it and you denaturate all the proteins. For Western blots, you treat the proteins with detergents, but also for immunofluorescence, sometimes you use ethanol. So in all of these cases, you denaturate the protein, the epitopes might change a lot, and therefore there is very much an issue if an antibody actually works. So what we have said then is that we try to have two antibodies binding to the same target, and then we compare the results from these two antibodies, and then we call them paired antibodies. And in this case, we know that what we are seeing is actually correct. But there are thousands and thousands of publications where antibodies, not from us, I hope, but from others, where antibodies have been shown, well, they have claimed that it worked, that it binds to a target, but actually it's cross-reactivity. So your question is very important, and especially then for applications where you denaturate the proteins, you have to be very careful, and just slight modifications might slightly change the epitopes, and then an antibody that works fine might not work. So, and this is something that each researcher has to be aware of and take precautions. Sorry for answering this very long, but this is a very important issue with antibodies. They are notoriously cross-reactive.
That's very insightful, detailed response. So that's great. One more participant, Dr. Asif, we want to, he's appreciating and congratulating you for your vision of past 20 years of HPA, what you have already shown, and he's wondering whether you also have make the plan for next 20 years. And he's asking for your vision for the HPA project for the next 20 years. So before you answer, I must say, Asif, I am seeing Professor Matthias for last several years, at least decade, and he has been showing his plan for next several years, And we have really exactly seen the date of publication and year of the same atlas coming in Science the way he has projected, 10 years ago. He came to one meeting in CCMP Hyderabad in 2009. At that same year, I joined India. And he showed his plan for at least next 5-6 years, and we followed exactly same publications came over the year. So I'm sure he has made the plan for 20 years, but he might be revealing us now. What is the plan?
Yeah. So thank you for that question. So it is interesting that the original application 20 years ago to the Wallenberg Foundation very much actually outlined what we have done in these years. I think we've done a little bit more than we thought we would do. So we're very proud of that. And we're of course very proud that we have almost 10 publications in Science. And actually, maybe I should mention that tomorrow there will be an article in Nature on single cell variability. So we're very proud of that from the group. But for the next, we are not planning for 20 years, but we just got funding for another 5 years from the Wallenberg Foundation. And what we are moving into then is the single cell analysis. We're also moving a lot more into the precision medicine but we're also trying to develop drugs using this knowledge that we have achieved. But our determination to put all the data open access without restrictions is still there. And we hope then to be able to continue the fantastic collaborations with Indian scientists that we are so happy and has been so successful for us in the past. So thank you very much. And I guess thank you also for listening.
No, thank you so much for this very wonderful sharing experience with us. And HP has become such an important resource for whole scientific community that with your accomplishments, I think whole scientific community is enjoying those resources. So I think we're all very glad to know that your support funding has been extended and you'll be now moving more into precision medicine area and looking at cell type atlas. I think we can take maybe one last question if somebody wants to. I think I saw some more questions on the forum here. Dr. Sudarshan, you want to ask a question? I think you have posted something.
Yes,
Go ahead.
I wanted to know, some of the proteins are evolutionarily conserved. So how do you select a host to generate antibodies against them?
So we have developed some software where for every protein that we want to make an antibody, we actually compare that sequence with all the other 20,000 protein-coded genes and proteins. And then we select out regions which are the most unique for that protein. And this is one of the reasons why we are not using full-length proteins, because if you have full-length proteins that you make antibodies to, there are always regions, just like you're saying, that are homologous between different proteins, and then you get cross-reactivity. So, but sometimes this is very difficult. There are proteins which are very, very similar. For example, there are globin proteins which only differ by one amino acid, and obviously it is very hard then to get a specific antibody for the two variants. So then in those cases, we actually use the antibody and we say that it actually binds to two very similar proteins, and we say that in the protein atlas. But this is of course a challenge, but it also is one of the reasons why we have decided to work with protein fragments, recombinant protein fragments, and not full-length proteins to make the antibodies.
I think one last question on the forum we have, from one participant, Dr. Ashok, how to differentiate the paralogs, which could it be possible to develop antibodies for same?
Is the question how, can you repeat that question, please?
Yeah, how to differentiate the paralogs? Could it be possible to develop antibody for same? Dr. Ashok, do you want to speak and
Yes, sir. Yes, sir. Sir, Hello, thank you, sir, for wonderful talk. My question is that I am working on protein which have 14 different paralogs, and getting the antibody for that is very challenging task at present for me. And so I need your guidance. Could it is possible to develop some antibody, because there are 14 different parallel nucleotide level. They are more than 90% similarity. They are shown.
Yes, so that's again sort of related to the question earlier on, and we have tried. I mean, it is very, if proteins are very similar and protein isoforms are similar, it is of course very hard to make antibodies, and we have focused then on making antibodies that are sort of directed to a representative protein from certain gene, and we've not worked so much with paralogs or isoforms, as I said earlier. This is of course a major challenge for the future, and something that I urge everybody to work on, and that is of course the question: how important is it with isoforms in human biology? And that is of course something that we don't really know. I mean, we know it's important, but we don't know how much of the complex human biology depends on the isoforms.
Sir, I have one, like I need your guidance. Could it is possible to any other proteomic techniques we could differentiate them? Because I found interesting finding, even they are similar in nucleotide level, but they have different functions. Even they have differential expression level in the same in the cancer. So there are any techniques like other methodologies so we utilize in proteomics and differentiate their expression?
I'm not sure actually. Maybe you can you actually send me an email about that, and we can actually have an email conversation, because I didn't really understand the question. But is that possible maybe? So,
Okay, sir. Sure.
I can have a connection with you, so that will be, we can just take one last comment here. One of the student feels that you are heading many startups. Do you have some startup in the precision medicine area?
Yeah. So I have actually started 25 biotech companies. 4 of them are on the stock market. And we are actually, these startup companies is now doing 11 clinical trials in humans right now. So I have been working a lot with applied work. 4 of these are in the field of precision medicine. So we have one company which is actually trying to work towards targeted proteomics, actually, using recombinant fragments and so on. We have a technology that we published together with Matthias Mann in Germany called PRESTs, which are sort of stable isotopes. And then we have another company which is actually working on trying to develop an assay for metformin treatment of diabetes and so on. So I am very, very devoted to basic research, but as you can hear, I'm also very fond of the entrepreneurial side of biotech, and that's why we have started 25 different startups. So yeah.
So just to add on this, I want to extend this question, like what are the areas you still feel that are uncovered? Like we have opportunity, we can grab those opportunity in field of precision medicine.
Well, I think we are in the most exciting era in human biology ever. I think the new technologies that we have, both the mass spec, the antibody-based profiling, the different omics technologies, they all are then directed towards a quantitative, data-driven life science. And I think with the competence that we have, for example, in India, I think this new era of life science should suit you very well, where data is shared, and then the secret is to be able to analyze the data in the best way. But I do think that, as I said, to get a holistic view of human biology and diseases is, of course, the goal. And in order to do that, we need quantitative methods which are very sensitive, both in the protein field, but also for metabolites and so on. I think we have the next 10 years will probably be the most exciting in life science ever, actually.
Great.
Thank you so much, sir. And we also invite you in India for opening any startup.
No, great. I think he will definitely visit sometime when the situation improves. Looks like participants don't want to let you go, but I think we have to. We understand you are very busy. I'll just close with Dr. Surekha Jingde's remark. I think she's also in the audience right now. Surekha, I think you know her well. She is one of the Indian key scientists of Proteome. She was the past president of PSI. So she mentioned that she enjoyed visiting HPA site. It is amazing to hear from you directly about the challenging work undertaken by your team under the leadership. Many congratulations. So I think on behalf of the entire workshop, IIT Bombay side, and all the Indian scientists who are here, we are very thankful to Professor Matthias Ullen for this wonderful lecture and sharing your very candid views about several issues and several opportunities which people have in this area. Thank you very much, Professor Matthias, and we look forward to have further interactions with you.
Yeah, thank you so much for being with us. I'm overawed to really hear it directly from you, all that I see on the HPA portal. Thank you so much.
Great. All right. So we will close this session now. With that,